my sort of preliminary dissertation work on the genomic population structure and its sort of implications for the evolution of freshwater tolerance in an invasive cnidarian, Cordylophora caspia. And this work has been done in conjunction with my advisor, Dr. Pauline Cartwright, and our collaborator from Wheaton College in Illinois, Nadine Roaring. And so just, I mean, I don't think any of us would be in this room listening to these talks if we weren't at least somewhat interested in questions such as, how does the evolutionary history of an organism affect both its current distribution and its invasive potential as a species? And for a few reasons, my colleagues and I think that this hydroid, Cordylophora, is a great system for studying questions like this. So, unlike many of its close relatives, which are either obligately freshwater or are marine, it inhabits a very wide range of salinities, and I'll talk a little bit more about that specifically. And notably for its invasion biology, it is a highly durable, almost cyst-like stage that makes it super easily transported in like ballast water, for example. And one of the things we enjoy about it is that it's pretty easy to collect and to grow different genotypes in our laboratory. So the current distribution of Cordylophora isn't even completely summed up in these pictures. It's pretty much found globally throughout the world. It was a native of the Ponto-Caspian region and has since spread throughout, pretty much every estuary system in Europe as well as the United States, and notably the freshwater systems that are connected to those estuary systems. So for example, in the United States, it's found throughout the southeast United States, such as in the Chesapeake Bay, some of the estuaries on the west coast, and in the Great Lakes, and even in some of those Finger Lakes in upstate New York. And this is really weird because other hydrozoans, such as Hydra, are just completely freshwater, and it comes from marine ancestors. So most other hydrozoans are completely marine. So previous phylogenetic work has indicated that it's not just that it's just one big species where every individual is euryhaline and could in theory be found anywhere. Rather, it was previously discovered that it is comprised of three different subspecies or clades, so it's probably a species complex of these three clades, one of which is this entirely freshwater group. One is a clade that contains colonies collected in both freshwater and salty places, and for the purposes of our work, we consider freshwater really quite freshwater, so basically 0.5 parts per thousand or less salinity, and then we have this clade of completely salty individuals. So this has led us to hypothesize that since its closest relatives, and indeed most hydrozoans are marine, that somewhere along this branch, the species involved the ability to tolerate things less than marine salinities, and on this branch, it actually lost the ability to tolerate anything greater than freshwater, so it became an obligate freshwater group here. And this is pretty remarkable since that freshwater, or even a couple parts per thousand, represents a pretty rough physiological barrier for lots of these marine invertebrates. And so this is a really well-sampled phylogeny, including some samples from pretty much around the world, but it's lacking, particularly in samples that systematically go up and down an estuary and collect samples from the length of that estuary. And estuaries are of course, sort of interesting places to study evolution because they encompass a range of salinities, both, of course, across their length as you get further or closer to the ocean, and certainly on like a daily tidal basis or even an annual basis as well. So sort of an interesting environment for adaptation to take place. And so our main question is, if we sample up and down different estuaries, where will all of these estuaries fall within the framework of the original phylogeny? Will they all, for example, be each other's closest relatives in a given estuary and probably be part of one of the two clades that contains brackish members? Or is an estuary going to be partitioned such that, for example, the freshwater individuals fall in this freshwater clade and vice versa? So those are the sort of two competing hypotheses. And once we look at a phylogenetic framework for this question, especially if it turns out that they're all closely related in a given estuary. We also want to know that in a given estuary, is there evidence of population structure along an estuary? So does it appear that there are any salinity barriers that present a gene flow barrier for this species? So our sampling scheme, so far my work includes 2 different estuaries, and essentially what we did for each of them is collected multiple colonies of Cordylophora at multiple different salinity levels throughout a given system, and just sort of based on the types of sampling we were able to get, we also happened to get populations or individuals from a population, further down the coast in this case, which might help us disentangle the effect of distance versus salinity on some of the genetic diversity measures. And in this case, we captured a range from pretty much freshwater to 7 points per thousand, which is again a wide range for some of these aquatic invertebrates. And likewise, in the Columbia River estuary in Washington and Oregon, almost the exact same thing. Multiple sites along an estuary, multiple colonies from each, a sampling site for a little further down the coast, and we tried to encompass as much a range as possible of salinity as we could. So our methodology was, once again, to sample, as I just talked about, to go ahead and just old school Sanger sequence the markers that were involved in the original phylogeny, just to see where our samples fell out in that exact context. We generated and sequenced a RAD-seq library, created de novo loci for population structure analyses, and I doubt I'll have time to talk too much about it, but if you want to compare methods or something, totally just come find me any other time at this meeting. And because we had some issues, shall we say, with prepping some of those libraries, this sort of unintentionally gave us the ability to examine how the number of loci included and the depth of coverage would affect any of those population structure results that we came across. So, right off the bat, our estuary samples are in red and blue in the next couple of phylogenies I'm going to show. This is my 16S plus Co1 phylogeny. We also have one that includes the 28S nuclear marker. It didn't have quite as good sampling and pretty much produced the exact same result, so I'm showing you this one. And what we find is that the topology is pretty much exactly consistent with the original phylogeny, in that we recovered 3 distinct clades. And notably, most samples from the 2 estuaries, even though they are 2 different estuary systems, and in theory could have been invaded by 2 separate invasions of the species, all fall in this clade that has pretty much maybe 1 or 2 nucleotide differences in this 16S Co1 haplotype. And notably, some freshwater colonies, so that is again less than 0.5 parts per thousand, fall amongst this previous just brackish higher salinity clade, so I'm going to relabel that guy mixed, question mark. And so you can see there's kind of little evidence that there's partitioning of the different clades along an estuary. However, we did note that there is a little bit of color going on in this previously only mixed group, where we note that sort of the most saline population from the San Francisco Bay estuary at 7 parts per thousand falls in a slightly different clade than the other ones, and also an oddball sample from the Columbia River estuary as well, which I'll bring up again towards the end. So, we used a RAD-seq loci, since it did pretty much turn out that they're a sort of undifferentiated looking clade, to see if they're adding more loci, looking at a little bit of a finer scale, would recover any sort of population structure. And I'm sure many of you in this room do this kind of thing and are familiar, but each of the vertical bars represents one of the individual colonies, and each color represents a population that is in theory, and Hardy-Weinberg and stuff, that the structure software has uncovered. And the color, how much of the bar is colored a certain color, is proportional to the probability that a given individual is in that color population. And notably, sort of the overall result is that, if you squint and look at it, preliminarily it looks like, again, that highest salinity population in California, so 7 parts per thousand, is sort of its own distinct group that is largely made up of what I'll call the blue population, and that this result was not super dependent on whether or not we had 12,000 loci or just, sadly, 12. And we also, just to help disentangle the effect of physical distance and salinity, I guess what you'd call salinity distance. We also just ran a Mantel test on genetic versus physical distance, which revealed some trend, as you might expect, but no significant correlation between the two. So we think that some of this differentiation, at least, is due, in fact, to salinity differences. And to just put it in context, again, with the map of this estuary, again, highest salinity population appears to be made up predominantly of members of one population, and that sort of within the rest of the estuary, there's not that much differentiation, and so this site was actually in the Los Angeles area, so that's in Irvine, the water treatment plant there. That does not appear to be its own separate invasion, even though it's pretty much all the way down its coast. We find a pretty similar pattern in Oregon and Washington. We find that our highest salinity population is a pretty distinct genetic population, and again, not dependent on number of loci or amount of coverage that those loci have. And so this is our population in Garibaldi, Oregon, and it appears to be its own separate population. Mantel test, again, did not reveal a particularly significant correlation between genetic and physical distance. But to put it in context with the map, Garibaldi is that sample that is both far away and the most salty. So in this case, it is harder to disentangle that question of, is it different just because it's far away, or is it different because it has a pretty different salinity regime? What's also pretty clear, though, is that within the Columbia River, so these 4 samples are collected basically down the river from each other and span 3.5 parts per thousand to pretty much completely freshwater, there's not that much differentiation between them. So it looks like there's plenty of potential gene flow, even between 4 parts per thousand and like 0 parts per thousand, which again is pretty rough stuff for your traditional aquatic invertebrate. So just to look at these examples in a framework that did not rely quite as heavily on assumptions of Hardy-Weinberg and stuff like that, I did some discriminant analysis of principal components, which basically tries to tell clusters apart based on variables that are made up of, in this case, combinations of the contribution of different loci across those populations. And what we find is that, sort of in contrast to what I found in my structure analysis, the California sites are all pretty much overlapping. Oregon does sort of mirror its population structure results, and you have that Garibaldi site coming out separately. So to conclude, in the San Francisco Bay estuary, there's some evidence for a break in gene flow, which separates that highest salinity population from the rest. Pretty much the same situation in Oregon, although isolation by distance cannot be ruled out. In either case, it doesn't look like within the estuaries themselves there's too much of a barrier to gene flow. And future work, we have more plates of rad sequencing coming down the pipe. We're going to do more intermediate, finer scaled sampling, comparison with what geographic structure looks like when we have no salinity differences, such as across the Great Lake. And we've also done collecting up and down the Chesapeake Bay to contrast it with this story here. And so I'd like to thank Dr. John Kelly and Jenny Hackett at the University of Kansas for their expertise in helping with library preps and sequencing and funding sources such as NSF, a Sigma Xi grant in native research, and my department for continued support and letting me go collect. All right, thank you.